Enzyme kinetics is the study of the chemical reactions that are catalyzed by enzymes. In enzyme kinetics, the reaction rate is measured and the effects of varying the conditions of the reaction are investigated. Studying an enzyme's kinetics in this way can reveal the catalytic mechanism of this enzyme, its role in metabolism, how its activity is controlled, and how a drug or an agonist might inhibit the enzyme. Enzymes are usually protein molecules that manipulate other molecules, the enzyme's substrates. These target molecules bind to an enzyme's active site and are transformed into products through a series of steps known as the enzymatic mechanism E plus SSS asterisk EP plus P. These mechanisms can be divided into single substrate and multiple substrate mechanisms. Kinetic studies on enzymes that only bind one substrate, such as triose-phosphate isomerase, aim to measure the affinity with which the enzyme binds this substrate and the turnover rate. Some other examples of enzymes are phosphofructokinase and hexokinase, both of which are important for cellular respiration. When enzymes bind multiple substrates, such as dihydrofolate reductase, enzyme kinetics can also show the sequence in which these substrates bind and the sequence in which products are released. An example of enzymes that bind a single substrate and release multiple products are proteases, which cleave one protein substrate into two polypeptide products. Others join two substrates together, such as DNA polymerase linking a nucleotide to DNA. Although these mechanisms are often a complex series of steps, there is typically one rate-determining step that determines the overall kinetics. This rate-determining step may be a chemical reaction or a conformational change of the enzyme or substrates, such as those involved in the release of product from the enzyme. Knowledge of the enzyme's structure is helpful in interpreting kinetic data. For example, the structure can suggest how substrates and products bind during catalysis, what changes occur during the reaction, and even the role of particular amino acid residues in the mechanism. Some enzymes change shape significantly during the mechanism. In such cases, it is helpful to determine the enzyme structure with and without bound substrate analogues that do not undergo the enzymatic reaction. Not all biological catalysts are protein enzymes. RNA-based catalysts such as ribozymes and ribosomes are essential to many cellular functions, such as RNA splicing and translation. The main difference between ribozymes and enzymes is that RNA catalysts are composed of nucleotides, whereas enzymes are composed of amino acids. Ribozymes also perform a more limited set of reactions, although their reaction mechanisms and kinetics can be analyzed and classified by the same methods. General principles. The reaction catalyzed by an enzyme uses exactly the same reactants and produces exactly the same products as the uncatalyzed reaction. Like other catalysts, enzymes do not alter the position of equilibrium between substrates and products. However, unlike uncatalyzed chemical reactions, enzyme-catalyzed reactions display saturation kinetics for a given enzyme concentration and for relatively low substrate concentrations. The reaction rate increases linearly with substrate concentration. The enzyme molecules are largely free to catalyze the reaction, and increasing substrate concentration means an increasing rate at which the enzyme and substrate molecules encounter one another. However, at relatively high substrate concentrations, the reaction rate asymptotically approaches the theoretical maximum. The enzyme active sites are almost all occupied and the reaction rate is determined by the intrinsic turnover rate of the enzyme. The substrate concentration midway between these two limiting cases is denoted by Km. The two most important kinetic properties of an enzyme are how quickly the enzyme becomes saturated with a particular substrate, and the maximum rate it can achieve. Knowing these properties suggests what an enzyme might do in the cell and can show how the enzyme will respond to changes in these conditions. Enzyme assays. Enzyme assays are laboratory procedures that measure the rate of enzyme reactions. Because enzymes are not consumed by the reactions they catalyze, enzyme assays usually follow changes in the concentration of either substrates or products to measure the rate of reaction. There are many methods of measurement. Spectrophotometric assays observe change in the absorbance of light between products and reactants. Radiometric assays involve the incorporation or release of radioactivity to measure the amount of product made over time. Spectrophotometric assays are most convenient since they allow the rate of the reaction to be measured continuously. Although radiometric assays require the removal and counting of samples, they are usually extremely sensitive and can measure very low levels of enzyme activity. An analogous approach is to use mass spectrometry to monitor the incorporation or release of stable isotopes as substrate is converted into product. The most sensitive enzyme assays use lasers focused through a microscope to observe changes in single enzyme molecules as they catalyze their reactions. These measurements either use changes in the fluorescence of cofactors during an enzyme's reaction mechanism, or of fluorescent dyes added onto specific sites of the protein to report movements that occur during catalysis. These studies are providing a new view of the kinetics and dynamics of single enzymes, as opposed to traditional enzyme kinetics, which observes the average behavior of populations of millions of enzyme molecules. An example progress curve for an enzyme assay is shown above. The enzyme produces product at an initial rate that is approximately linear for a short period after the start of the reaction. As the reaction proceeds and substrate is consumed, the rate continuously slows. To measure the initial rate, enzyme assays are typically carried out while the reaction has progressed only a few percent towards total completion. The length of the initial rate period depends on the assay conditions and can range from milliseconds to hours. However, equipment for rapidly mixing liquids allows fast kinetic measurements on initial rates of less than 1 second. These very rapid assays are essential for measuring pre-steady-state kinetics, which are discussed below. Most enzyme kinetics studies concentrate on this initial, approximately linear part of enzyme reactions. However, it is also possible to measure the complete reaction curve and fit this data to a nonlinear rate equation. This way of measuring enzyme reactions is called progress curve analysis. This approach is useful as an alternative to rapid kinetics when the initial rate is too fast to measure accurately. Single substrate reactions. Enzymes with single substrate mechanisms include isomerases such as triose-phosphate isomerase or bisphosphoglycerate mutase, intramolecular lyases such as adenylate cyclase and the hammerhead ribozyme, and RNA lyase. However, some enzymes that only have a single substrate do not fall into this category of mechanisms. Catalase is an example of this, as the enzyme reacts with the first molecule of hydrogen peroxide substrate, becomes oxidized and is then reduced by a second molecule of substrate. Although a single substrate is involved, the existence of a modified enzyme intermediate means that the mechanism of catalase is actually a ping-pong mechanism, a type of mechanism that is discussed in the multi-substrate reactions section below. Mccoys kinetics as enzyme catalyzed reactions are saturable. Their rate of catalysis does not show a linear response to increasing substrate. If the initial rate of the reaction is measured over a range of substrate concentrations, the reaction rate increases as S increases, as shown on the right. However, as S gets higher, the enzyme becomes saturated with substrate and the rate reaches Vmax, the enzyme's maximum rate. The Mccoys kinetic model of a single substrate reaction is shown on the right. There is an initial bimolecular reaction between the enzyme E and substrate S to form the enzyme-substrate complex S, but the rate of enzymatic reaction increase with the increase of the substrate concentration up to a certain level, but then more increase in substrate concentration does not cause any increase in reaction rate, as there no more E remain available for reacting with S, and the rate of reaction become dependent on S, and the reaction become unimolecular reaction. Although the enzymatic mechanism for the unimolecular reaction can be quite complex, there is typically one rate-determining enzymatic step that allows this reaction to be modeled as a single catalytic step with an apparent unimolecular rate constant kCat. If the reaction path proceeds over one or several intermediates, kCat will be a function of several elementary rate constants, whereas in the simplest case of a single elementary reaction it will be identical to the elementary unimolecular rate constant K2. The apparent unimolecular rate constant kCat is also called turnover number and denotes the maximum number of enzymatic reactions catalyzed per second. The Mccoys-Constant equation describes how the reaction rate V0 depends on the position of the substrate binding equilibrium and the rate constant K2. With the constants, this Mccoys-Constant equation is the basis for most single substrate enzyme kinetics. Two crucial assumptions underlie this equation. The first assumption is the so-called quasi-steady-state assumption, namely that the concentration of the substrate-bound enzyme changes much more slowly than those of the product and substrate, and thus the change over time of the complex can be set to zero. The second assumption is that the total enzyme concentration does not change over time, thus a complete derivation can be found here. The Mccoys constant Km is experimentally defined as the concentration at which the rate of the enzyme reaction is half Vmax, which can be verified by substituting S equals Km into the Mccoys-Constant equation, and can also be seen graphically. If the rate-determining enzymatic step is slow compared to substrate dissociation, and are sometimes referred to as kinetically perfect enzymes. Direct use of the Mccoys-Constant equation for time-course kinetic analysis. The observed velocities predicted by the Mccoys-Constant equation can be used to directly model the time-course disappearance of substrate and the production of product through incorporation of the Mccoys-Constant equation into the equation for first-order chemical kinetics. This can only be achieved, however, if one recognizes the problem associated with the use of Euler's number in the description of first-order chemical kinetics, i.e., E-K is a split constant that introduces a systematic error into calculations and can be rewritten as a single constant which represents the remaining substrate after each time period. In 1983, Stuart Beal derived a closed-form solution for the time-course kinetics analysis of the Mccoys-Constant mechanism. The solution, known as the Schnell-Mendoza equation, has the form where W is the Lambert W function, and where F is this equation is encompassed by the equation below, obtained by Berber and Santos 283, which is also valid when the initial substrate concentration is close to that of enzyme, where W is again the Lambert W function. Linear plots of the Mccoys-Constant equation. The plot of V versus S above is not linear, although initially linear at low S. It bends over to saturate at high S. Before the modern era of nonlinear curve fitting on computers, this nonlinearity could make it difficult to estimate Km and Vmax accurately. Therefore, several researchers developed linearizations of the Mccoys-Constant equation, such as the Lineweaver-Burk plot, the Eadie-Hofstee diagram and the Haynes-Wolff plot. All of these linear representations can be useful for visualizing data, but none should be used to determine kinetic parameters, as computer software is readily available that allows for more accurate determination by nonlinear regression methods. The Lineweaver-Burk plot or double reciprocal plot is a common way of illustrating kinetic data. This is produced by taking the reciprocal of both sides of the Mccoys-Constant equation, as shown on the right. This is a linear form of the Mccoys-Constant equation and produces a straight line with the equation y equals mx plus c with a y-intercept equivalent to 1 v max and an x-intercept of the graph representing -1 km. Naturally, no experimental values can be taken at -1 s. The lower limiting value 1 s equals 0 corresponds to an infinite substrate concentration, where 1 v equals 1 v max is shown at the right. Thus, the x-intercept is an extrapolation of the experimental data taken at positive concentrations. More generally, the Lineweaver-Burk plot skews the importance of measurements taken at low substrate concentrations and thus can yield inaccurate estimates of Vmax and Km. A more accurate linear plotting method is the Eadie-Hofstee plot. In this case, V is plotted against V, S. In the third common linear representation, the Haynes-Wolff plot, S, V is plotted against S. In general, data normalization can help diminish the amount of experimental work and can increase the reliability of the output, and is suitable for both graphical and numerical analysis. Practical significance of kinetic constants. The study of enzyme kinetics is important for two basic reasons. Firstly, it helps explain how enzymes work, and secondly, it helps predict how enzymes behave in living organisms. The kinetic constants defined above, Km and Vmax, are critical to attempts to understand how enzymes work together to control metabolism. Making these predictions is not trivial, even for simple systems. For example, oxaloacetate is formed by malate dehydrogenase within the mitochondrion. Oxaloacetate can then be consumed by citrate synthase, phosphenol pyruvate carboxykinase or aspartate aminotransferase, feeding into the citric acid cycle, gluconeogenesis or aspartic acid biosynthesis, respectively. Being able to predict how much oxaloacetate goes into which pathway requires knowledge of the concentration of oxaloacetate as well as the concentration and kinetics of each of these enzymes. This aim at predicting the behavior of metabolic pathways reaches its most complex expression in the synthesis of huge amounts of kinetic and gene expression data into mathematical models of entire organisms. Alternatively, one useful simplification of the metabolic modeling problem is to ignore the underlying enzyme kinetics and only rely on information about the reaction network's stoichiometry, a technique called flux balance analysis. Mccoys-Constant kinetics with intermediate. One could also consider the less simple case where a complex with the enzyme and an intermediate exists, and the intermediate is converted into product in a second step. In this case we have a very similar equation but the constants are different. We see that for the limiting case. Thus when the last step from EI to E plus P is much faster than the previous step, we get again the original equation. Mathematically we have then in multi-substrate reactions. Multi-substrate reactions follow complex rate equations that describe how the substrates bind and in what sequence. The analysis of these reactions is much simpler if the concentration of substrate A is kept constant and substrate B varied. Under these conditions, the enzyme behaves just like a single substrate enzyme in a plot of E by S, gives apparent Km and Vmax constants for substrate B. If a set of these measurements is performed at different fixed concentrations of A, these data can be used to work out what the mechanism of the reaction is. For an enzyme that takes two substrates A and B and turns them into two products P and Q, there are two types of mechanism: ternary complex and ping-pong. Ternary complex mechanisms. In these enzymes, both substrates bind to the enzyme at the same time to produce an EAB ternary complex. The order of binding can either be random or substrates have to bind in a particular sequence. When a set of V by S curves from an enzyme with a ternary complex mechanism are plotted in a Lineweaver-Burk plot, the set of lines produced will intersect. Enzymes with ternary complex mechanisms include glutathione S-transferase, dihydrofolate reductase and DNA polymerase. The following links show short animations of the ternary complex mechanisms of the enzymes dihydrofolate reductase and DNA polymerase. Ping-pong mechanisms as shown on the right. Enzymes with a ping-pong mechanism can exist in two states, E and a chemically modified form of the enzyme E*. This modified enzyme is known as an intermediate. In such mechanisms, substrate A binds, changes the enzyme to E** by, for example, transferring a chemical group to the active site, and is then released. Only after the first substrate is released can substrate B bind and react with the modified enzyme, regenerating the unmodified E form. When a set of V by S curves from an enzyme with a ping-pong mechanism are plotted in a Lineweaver-Burk plot, a set of parallel lines will be produced. This is called a secondary plot. Enzymes with ping-pong mechanisms include some oxidoradicases such as thioredoxin peroxidase, transferases such as acylneuraminate cytadilyl transferase and serine proteases such as trypsin and chymotrypsin. Serine proteases are a very common and diverse family of enzymes, including digestive enzymes, several enzymes of the blood clotting cascade and many others. In these serine proteases, the E** intermediate is an acyl enzyme species formed by the attack of an active site serine residue on a peptide bond in a protein substrate. A short animation showing the mechanism of chymotrypsin is linked here.